Hi everyone, welcome back to Chapter 11. Today we're going to look at mutations, what they are, how they happen, and how cells repair them. Our goals for section 11.5 are to compare point and frameshift mutations, differentiate missense, nonsense, and silent mutations, understand light versus dark repair, explain how mutagens work, explore how the Ames test detects carcinogens, and finally identify examples of different mutation types from DNA sequences. So keep these objectives in mind. They'll help guide what you should take away from this lecture. A mutation is a heritable change in DNA sequence, and these changes can alter how genes are expressed and how proteins function. The altered form is called the mutant, whereas the normal version is known as the wild type. Mutations can happen spontaneously, just from replication errors, or be induced by mutagens, and these are things like chemicals or radiation. While most mutations are neutral or harmful, occasionally a mutation will be beneficial, giving an organism some kind of survival advantage. And we categorize mutations by the type of DNA change. Point mutations involve a single base change. Insertions and deletions add or remove nucleotides. When insertion or deletion is not in a multiple of 3, we call it a frame shift. Frame shifts will alter the reading frame and often lead to non-functional proteins because every codon downstream of the mutation will change. This checkpoint asks, what type of mutation occurs when a gene has 2 fewer nucleotides in its sequence? So imagine this is the normal sequence, and 2 nucleotides, it looks like these 2, are going to be deleted. That means everything downstream will be changed. So this shifts the reading frame. The reading frame used to be like this. Now, instead of we have CUU is normal, now it's shifted. So the reading frame has been shifted. Everything downstream of the mutation will be changed, all those downstream codons, and this will often produce a non-functional protein. So let's look at how these mutations affect protein function. And all of these are point mutations where there is a change in one base pair. In silent mutations, there's a base change, but the same amino acid is still formed. There's no functional change. In a missense mutation, one amino acid is replaced by another. Sometimes this is harmless if the amino acids are very similar, but if the new amino acid is chemically different, it can change protein folding or function. Within this missense mutation category, we have conditional mutations. And in conditional mutations, the missense effect only shows under certain conditions. For example, a temperature-sensitive enzyme that folds normally at 1 temperature, but misfolds at high temperatures. We also have nonsense mutations, and these introduce a stop codon too early, producing a shortened or truncated protein. So these mutations really depend on the chemical difference of amino acids or the position of the change in the base, and some missense mutations are conditional, others can be beneficial. Let's take a look at our different mutations more closely. Here we have point mutations, and down here we're going to look at frameshift mutations. Let's start with our point mutations, which is when we change a single base. This looks like my DNA, my wild-type DNA, my messenger RNA, and corresponding amino acid chain. Here are my codons. And if I look at my DNA versus my messenger RNA, they look very similar in sequence. So the DNA must be my sense strand or coding strand, right? These are almost identical in sequence, except I have U's instead of T's. And in my first mutation, looks like I used to have a GTA, that's my normal DNA. It has been mutated to GTT. My codon used to be GUA, now it's GUU. Luckily, the resulting amino acid is still the same, so there's no effect on the protein sequence or function. In my second example, I have a missense mutation, and this time it looks like the mutation is happening around here. Instead of CCC, I have ACC in my DNA sequence, so that changes my codon from CCC to ACC. Unfortunately, this time it does change my amino acid, and I wasn't sure how different these are. So if the two amino acids were very similar, it might not change the function of your protein. But if these are very different, it could. So here's proline. This is a non-polar, rigid, ring-shaped amino acid versus threonine is a polar, very flexible amino acid. So that is a pretty big difference between the two amino acids. This likely will have a very different protein shape resulting from that change, that missense mutation, and it will likely affect the protein function. In the third point mutation, we have a nonsense mutation. So it looks like in that last one in my DNA, instead of TAC, I have TAG. Instead of my codon UAC, now I have UAG, and that is a stop codon. So that's going to result in an early stopping of my translation. So I'm going to have a truncated or shortened protein. Finally, going to frameshift mutations. These occur when you insert or delete a number of bases that is not a multiple of 3. So let's take a look at this example. So it looks like this was my normal reading frame, and they're going to delete these 2 bases. So T and A are removed. So everything is shifted to the left. Let's take a look. So now G, C, C, those are going to form a reading frame, a codon right here. Everything has been shifted. And we can see the result is everything from the mutation on downstream. All of those codons are different, and all of those resulting amino acids are different. That is going to lead to a very different protein, a non-functional protein. Our next checkpoint asks, what are the reasons a nucleotide change in a gene for a protein might not have any effect on the phenotype? So this could be a silent mutation where the altered codon still codes for the same amino acid, and that would be due to that degeneracy of the genetic code, where more than one codon can be encoding a single amino acid. Or the amino acid change might not affect the protein function if it occurs in a non-critical region of the protein. Alternatively, the new amino acid could have similar chemical properties as the previous or normal amino acid. For the second checkpoint question, is it possible for an insertion of 3 nucleotides together after the 5th nucleotide to produce a protein that's shorter than normal? Let's check it out. So here I gave an example of a wild type. Let's say this is my normal or wild type DNA. Next down here is my corresponding transcript, my messenger RNA, and let's say this is the normal reading frame. All right, so that's my normal reading frame, and I'm going to insert 3 nucleotides. I put in red after the 5th one. 1, 2, 3, 4, 5, so it's going to be right here. That's where I've inserted my new set of 3 nucleotides. And I'm going to change colors really quick. So here is my reading frame. It does not result in a frame shift because you can see these are still in the normal reading frame. There's a temporary disruption here, but everything reverts because we're doing an insertion that's a multiple of 3. So this could produce a protein that's shorter than normal, even though you think 3 nucleotides would equal 1 codon or 1 extra amino acid, if it introduces a stop codon, which it does right here. So here I have UAG. That means this would be the last amino acid, and then translation would stop. That would be the termination of translation right there. Our book has a great example of a real-world example of a beneficial mutation. So the CCR5 delta 32 deletion actually removes 32 base pairs from a gene called the CCR5 gene. So this happens to be a receptor that normally sits on the surface of T cells, and this normally allows HIV to enter these T cells. But in this mutation, people that are homozygous, that have 2 copies of this mutated gene, lack a functional CCR5 receptor, and they're resistant to infection, whereas heterozygotes, which have only 1 copy of this mutation, have partial protection. So this mutation likely became common in Northern Europe due to natural selection, maybe during smallpox or plague outbreaks, and today it's guiding new HIV drug research. So what causes these mutations? It could be spontaneous through DNA replication errors, but that is pretty rare because the error rate of DNA polymerase is something like 1 per a billion base pairs. So most commonly it's through mutagens, and mutagens can be chemicals or radiation. So this would be induced mutations. For chemical mutagens, we have many different types. Some are called nucleoside analogs if they are very similar in structure to one of our normal nucleosides, A, T, C, or G, and others can be modifiers that actually chemically modify one of our nucleosides. An example could be nitrous acid, so that would be a chemical modifier. We also have chemicals known as intercalating agents that distort our DNA helix because they kind of slide themselves into the helical structure, usually where the base pairs are hydrogen bonding. And these can often cause insertions or deletions because of that distortion in the shape of the DNA. And examples of intercalating agents include ethidium bromide or acridine orange. So let's take a closer look at those mutagens. We have two types here. At the top half, we have nucleoside analogs. These are mutagens that look just like one of our normal bases. So here is normal adenine nucleoside, and here is a mutagen, a nucleoside analog. This is 2AP, 2-aminopurine nucleoside. It looks just like A, but whereas A usually pairs with T, the AP analog pairs with C. So after a few rounds of replication, this will turn into a GC base pair, whereas it used to be AT in the wild type normal DNA. 5-bromouracil is another nucleoside analog that looks just like thymine. So they look very similar, except T usually pairs with A, right? But 5-bromouracil pairs with guanine. So also after a few rounds of replication, this will become a GC pair, whereas the wild type was TA. Down here, we have a different type of mutagen. These are chemical mutagens, and in particular, for this example, we have nitrous acid. Nitrous acid converts cytosine, so it chemically modified cytosine, forming uracil. So whereas we used to have some kind of CG pair, over time this will become UA, which actually becomes TA. It becomes a TA base pair. We also have mutagens called intercalating agents, and these include ethidium bromide or acridine orange that we use to stain nucleic acids. So here I can see acridine is inserting itself into my DNA double helix. It likes to insert itself between the base pairs. And what happens, or what can happen, is it introduces atypical or abnormal spacing between base pairs, and this can lead to a potential frameshift mutation. Exposure to radiation can also cause mutations. There are two types here. We have ionizing radiation, such as X-rays and gamma rays. These are very powerful rays that can cause DNA breaks in the backbone of DNA. We also have non-ionizing radiation, such as ultraviolet light, and that forms something called pyrimidine dimers, most often known or seen in the form of thymine dimers. So here we see ionizing radiation on the left. The effects are either single or double-stranded breaks in the backbone of the DNA, and this can be caused by very powerful X or gamma rays. On the right, we have non-ionizing radiation caused by UV light, UV light exposure, and this can cause pyrimidine dimers. Remember, the pyrimidines are C, U, and T, so we're looking at DNA, C or T are possible. We see it most commonly in thymines, where instead of base pairing to their complementary base in that double helix structure, they form covalent bonds with each other when they're right next to each other. So these thymines are covalently bonded, and that can stall, it can inhibit replication and transcription. It can also introduce frame shifts or point mutations. And our book has a great table of these different mutagens or mutagenic agents. We have our nucleoside analogs. Remember, these look just like one of our nucleotides or nucleosides, but they're not, and they change the base pairing. So these were point mutations. We also had that chemical that modified our base and converted cytosine to uracil. So that also results in a point mutation here. We had our intercalating agents like acridine orange or ethidium bromide. We talked about X and gamma rays that cause single or double-stranded breaks. I didn't talk about this, but it can also cause point mutations. And then our non-ionizing radiation like UV light that causes thymine dimers and can cause errors in replication or transcription or frame shift or point mutations. Our next checkpoint question asks, how does a base analog introduce a mutation? So we saw that a base analog resembles a normal nucleotide and can be incorporated into DNA during replication. It pairs incorrectly with the other bases, leading to a base substitution mutation in future rounds of replication. For the next question, how does an intercalating agent introduce a mutation? So intercalating agents like ethidium bromide or acridine orange, they insert themselves between the stacked DNA bases, and this will distort the DNA double helix and can cause insertions or deletions during replication, often producing those frame shift mutations. The third question asks, what type of mutagen causes thymine dimers? So we just saw that this was ultraviolet or UV radiation. This is a type of non-ionizing radiation that causes these thymine dimers, where two adjacent thymines covalently bond to each other instead of base pairing with their complementary base. And this will distort DNA structure and can block replication and transcription. So luckily, cells have several mechanisms to fix DNA damage. First, we have proofreading. Our DNA polymerases actually check their work along the way while they're making the daughter strand. Every few nucleotides, they'll go backwards and double check that the correct bases have been laid down, and they will remove mismatched bases if necessary. In mismatch repair, this occurs after replication has occurred. So basically, if anything was not caught, if there were any errors that were not caught by DNA polymerases, the other enzymes will jump in and correct these errors. To repair thymine dimers, there are two types of repair. We either have light repair, also known as photoreactivation, where an enzyme called photolyase will break the covalent bonds between those thymines, or dark repair, also known as nucleotide excision repair, where enzymes will remove the damaged segment. Humans only have dark repair. We don't have photolyase. So photoreactivation is absent in us and other mammals. And our book has a nice example of how bacteria repair those thymine dimers created by UV light. We have dark repair, also known as nucleotide excision repair, where that damaged segment is removed, and DNA polymerase I and ligase will replace with the correct nucleotides. In the light repair, we have that photolyase present in bacteria that allows photoreactivation to occur. Photolyase will break that covalent bond or those covalent bonds between the adjacent thymines, and now they can reform the hydrogen bonds with their corresponding bases. Our next checkpoint question asks, during mismatch repair, how does the enzyme recognize which is the new and which is the old strand, right, the template strand? So it turns out that in bacteria, the new strand is temporarily unmethylated immediately after replication. So the repair enzymes recognize the unmethylated strand as the new one and correct mismatches using the old or methylated strand as the template. So methylation, remember methyl groups look kind of like that. So there will be methyl groups on the old strand, but not the new strand at first. In the second question, they ask, what type of mutation does photolyase repair? We saw that photolyase repairs thymine dimers that are caused by UV light, caused by that non-ionizing radiation. And it uses visible light energy to break the covalent bond between thymine bases, and this process was called photoreactivation. So to study mutations, we can use different types of techniques. One is replica plating, and the other we'll talk about later is the Ames test. In replica plating, we will identify auxotrophs. These are mutants that have lost the ability to synthesize a nutrient. And this is in contrast to prototrophs, which are wild type and can grow on media called minimal media, where we don't have to add basic nutrients. So an example of an auxotroph is written as this, His minus. This means that this is a mutant, a type of mutant bacteria that can no longer make the amino acid histidine. In contrast, the wild-type version is His plus. That is a bacterium that can produce its own histidine. And this figure shows how replica plating works. So first, colonies are grown on a type of media called complete medium. And complete media has basically all the nutrients a microbe could want, including all the basic nutrients. And then we're going to take a velvet pad and kind of photocopy our colonies. So you touch your colonies and the colonies are transferred onto the velvet pad. You can see them here, those dots. And then you touch the velvet pad onto two different plates. Complete medium, medium containing the nutrient histidine, and a medium lacking that nutrient. Then we'll incubate for a couple of days, either 24 or 48 hours often, and we'll see that since we use the same velvet pad to stamp both plates, every colony corresponds to an identical colony on the other plate as long as they're in the same location. So this bacterium is identical to that bacterium. I'll put that one. Let's say that one is identical to that one. And the one they circled, they're identical. For some reason it grows here, but it doesn't grow here. It's missing. The reason for that is everything can grow if all the nutrients have been provided for the bacterium. So here we've provided histidine. So even if it was a mutant that could not make its own histidine, it can survive because we fed it histidine, right? We gave it histidine. It doesn't have to make it on its own. But on this minimal medium where we did not provide histidine, this bacterium could no longer grow, right? It can't make its own histidine and you did not supply it with histidine, so there is no growth. And you can go back to this first plate with all the nutrients, this complete medium, and pick that colony and study it further if you'd like to. Why are cells plated on a nutritionally complete plate? So that's a complete medium, in addition to nutrient deficient plates, these are called minimal media, when looking for mutants. So the complete plate, that complete medium, serves as a growth control. It confirms that the cells are alive and they're viable. Colonies that grow on the complete medium, but not on the nutrient deficient medium, are auxotrophic mutants. They're unable to synthesize the missing nutrient. So we saw earlier that we had histidine auxotrophs, so His minus auxotrophs, that were unable to produce their own histidine, so they could not grow when you did not supply it with histidine, but if you grow them on complete media, they can grow. That other test was the Ames test, and in the Ames test, we typically use Salmonella bacteria that are histidine auxotrophs. We use these bacteria to detect mutagenic chemicals or solutions. And some of these are carcinogenic. These could be potentially cancer-causing. So exposure to the mutagen should increase reversion rate in histidine auxotrophs. So it's almost like a double negative. His minus bacteria will become His positive if the chemical you're testing is a mutagen. And we also typically add some rat liver extract to the solution because we want to mimic the metabolism of chemicals in the human body. Sometimes something that is not itself a mutagen becomes mutagenic after it's metabolized by our liver. All right, so here's the Ames test. Here's my control over here. I'm working with His minus, right? His minus bacteria, and I'm exposing the rat liver extract to my Salmonella bacteria. Nothing should happen. There is some natural revertance, so some His minus become His positive just because of spontaneous mutations. And now I'm testing my chemical. All right, I want to see if my chemical is a mutagen, is a possible mutagen. So when I do the same thing this time, I see tons of His positive organisms, these mutants. So because of the high number of revertants, I know that this is a mutagen. This first checkpoint question asks, what mutation is used as an indicator of mutation rate in the Ames test? So we just saw the Ames test uses histidine auxotrophs of Salmonella bacteria, and reversion from a His minus mutant back to the His plus wild type indicates that a mutation has occurred. And the second question, why can the Ames test work as a test for carcinogenicity? So many carcinogens are also mutagens. They cause DNA mutations that can lead to cancer. So the Ames test measures how often a substance causes mutations in bacteria, making it a quick and expensive indicator of potential carcinogenic risk in humans. All right, let's recap 11.5. In the first learning objective, compare point mutations and frameshift mutations. We saw point mutations are a change in a single nucleotide base pair, and they can result in silent, missense, or nonsense effects, whereas a frameshift mutation is an insertion or deletion of bases that are not in multiples of 3, and this shifts the reading frame, altering all downstream codons, and usually produces a completely non-functional protein. For the next several ones, I'm going to use a table to describe the ones I've marked in red. So let's jump to the 5th one for now. We saw why the Ames test can be used to detect carcinogens. So we just mentioned that many carcinogens are also mutagens that damage DNA. So the Ames test measures the reversion rate in Salmonella histidine auxotrophs. Remember, from His minus to His plus. So a chemical that increases the mutation rate likely increases cancer risk in higher organisms like us. And that inclusion of the rat liver extract simulates mammalian metabolism to detect compounds that are activated in the body. The second learning objective asks us to basically look at the difference between missense, nonsense, and silent mutations. We saw missense mutations result in a different amino acid. It can or cannot affect protein function depending on how similar those amino acids are to each other. Nonsense mutations, we add a stop codon, right? A normal codon becomes a stop codon, resulting in a shortened protein. And in a silent mutation, that would be the one we would want to have if we had to have a mutation. We have the same amino acid put down because of that genetic code degeneracy. Our third learning objective asks us to compare light and dark repair. So light repair, or photoreactivation, used light energy, and photolyase was the enzyme involved that broke those thymine dimer bonds. In dark repair, there's no light needed. This is also known as nucleotide excision repair. And remember, an endonuclease would come in and cut out that damaged segment. DNA polymerase I and ligase would come in and repair that segment. For learning objective 4, how different mutagens act, these are just some examples. It's not all inclusive. So we talked about base analogs. Remember, these look like one of our normal bases, but they have different base pairs. We had base modifiers like nitrous acid that converted one base to another. We have intercalating agents like ethidium bromide that insert themselves between our DNA bases. And we have radiation such as UV light, that non-ionizing radiation that can cause thymine dimers. We also saw X-rays or gamma rays. Those are ionizing forms of radiation that can cause strand breaks in our backbone of our DNA. And for the final learning objective, analyze different sequences of DNA and give examples or identify examples. I put together some different sequences or different mutations that could be possible, and see if you can fill in these two columns here using our codon chart, our genetic code. The answer key will be in the next slide. Here is a key to that chart, and notice I put template strand in some of these rows, and these would be the corresponding mRNA codons. Take a look at your answers, double check to see if they were correct. And that takes us to the end of video 4. In our next video, we'll talk about gene transfer and how that increases genetic diversity in prokaryotes.